Is anyone speaking?
No, no, no. No one's speaking. So
Oh, okay.
I'm about to start the class. Yeah.
Oh, okay.
So am I audible and my screen is visible?
Yes.
Yeah. Okay. Cool. So hello everyone and yeah, welcome everyone to the week 7 of the TA session for the course Research Methodology in Natural Sciences. So yeah, so last week we have for the past 6 weeks we have had some uh, lectures and uh, from that we have had some recap of the lectures in the TA session and then we have been discussing some problems related to that. So this week it will be a continuation of that where I will be discussing um, where I'll be going through a general recap of whatever happened in the previous week's lectures and then we will be going over the problems of both week 6 and week 7. Yeah. So yeah, I'll start now. And one major disclaimer is that I suggest everyone in the call, everyone else in the call, to mute their audio as well as switch off their video. This is just to prevent any other interference in the call and just to protect the bandwidth of the other users in the lab, other users in the call. So I'll jump right into the recap. So once again, I'll just get my laser pointer on. Yeah. So last week, compared to last week as well as in week 6, so week 6 started off with some lectures which dealt off with standard error and we ended off with a series of lectures on measurement of proportion. Uh, the topic was a bit vague in last week's lectures, but half of it was covered in week 6 and the rest of measurement of proportion was covered in week 7. So that is where we'll be starting right off. We'll be covering a general recap of measurement of proportion and then we'll going on towards the topics of week 7. So measurement of proportion. So in the lectures of week 6 as well as week 7, the course instructor Dr. Soumitra Banerjee, he explained about the need of measurement of proportion, generally why it is used for. So measurement of proportion is a metric. It's a proportionatory metric which is commonly used for studying changes in nature or in science. So modern scientific materialism is based on the study of change and change from one form of any matter to another form. So during change, the population or certain traits are bound to change from one form to another. So keeping track of these changes is essential for studying the whole process as such. And how do we do that? One major method by which we track these changes is by using certain measurements of proportion. And one common example that we saw in the week's lectures was the occurrence of the white moth and the black moth. So before the Industrial Revolution, England had a huge population of white moths, which were sitting on which were sitting on lichens and other trees. And since lichens are white and moths are white, they were able to easily camouflage and they were able to avoid predation by birds. Birds were not able to capture their prey and eat them. But after the advent of after industrial revolution came into existence, there was a lot of production of smoke and soot and other carbon materials. So the deposition of this smoke and other microparticles on the lichens as well as the tree bark of the trees cause the discoloration of the barks into black color, and as a result, the moths were visible, were comparably visible to the environment. So as a consequence, the birds were able to target this white moths and then eat them. So as a result, what happened is to combat this predation prey situation, the white moths underwent a natural selection, after which most of the moths were black in color so that they could camouflage with the nature once again seamlessly and then avoid the predation. So there is a change in the number of population of white moths and black moths over a period of time. So the period in which this change has happened and the number from which this change has happened will lead to the study of a lot of evolutionary concepts. This was Darwin's theory. So that's how the measurement of proportion is brought into debate. So there are a lot of concepts, mathematical concepts being discussed in this concept of measurement of proportion. Mainly the result of measurement. So let's just if we just assume y as a result of measurement, let's just say if we just say a result of measurement as what is the when we measure the color of moths, if it's white or black, that is the result of measurement. So if we just assume zeros and ones to it, so that will be y. And if we want to take a event such as x, where x is the number of white moths in number of samples, or number of black moths in number of samples, that event would be the cumulative sum of all the result of measurements that you've done across n samples. So if x, let's just say we assign 1 to black moths and 0 to white moths. So if you want to calculate the number of black moths in a number of samples, so for each moth you'll be doing a measurement, and that will come under y, and the sum of all the measurements over n samples will constitute the event x. So eventually what will happen is the mean of this x event, number of ones and number of samples, will eventually come to the cumulative sum of the individual means of all the events that happen within all the, not all the events, all the results of measurements that come within the event. And subsequently the variance will also be the some number of events multiplied by the variance of each result of measurement, which will come down to around n into p into 1 minus p, where p is the weighted probability of each y. So y can have zeros and ones. So depending on the probability of ones and zeros that can have in nature, that will come under p, where p is the weighted probability. But weighted probability as in probability into number of those species plus probability into number of the other species. So that's what you called as weighted probability. And after that we came into the main picture, which is the measured proportion. And measured proportion would be described as uh, the number, the events, the parameter that we get in an event divided by the total number of events, and the mean of that event will always come under a normal distribution if we take lot of samples, just as we discussed in the central limit theorem, where if you take the mean of all the different samples that you take under a population, if you plot a distribution of the means, that will always come to a normal distribution which has a mean of the population as well, and the standard deviation divided by the having a standard error as the standard deviation of the plot distribution. And a common rule of thumb which was discussed whether the measured proportion will have a normal distribution was that if the number of is the probability of the events multiplied by the number of events is greater than 10, and vice versa, probability of the opposite event. uh, Amit Srinivastava, we will have a doubt session after this. I will come to your question during the doubt session. I'll first finish my recap of first, if that's okay with you. Yeah. So if p times n and 1 minus p times n is greater than 10, then the rule of thumb is followed that the distribution of the measured proportion will be approximately normal. And we also conclude that compared to the central limit theorem, where we need 25 samples as the minimum samples required to obtain a normal distribution, here they conclude that we need large number of samples. Apart from this being a criteria, p times n being greater than 10, we need other criteria such as large number of samples to achieve a higher confidence interval. And we continue with week 7's lectures, where we first see about box and whisker plot. So in last week's lectures we saw about error bars. So while plotting any data, we saw about how we plot mean and the standard error as the error bars, but that doesn't give you other information about the set of values. So to display all the other values that we have in a data, this box and whisker plot comes into place. And this is a common example of how a box and whisker plot looks like. So this region here, this is the box, and the small lines that we have here are the whiskers. So what does this box and whisker mean? So the whisker here, the terminal lines which are present here, those just detail the extremities of the data. So the lower line here describes the lower extreme of the data, and the upper line here describes the upper extreme of the data. So let's just say we have 10, 15 values and you try to arrange them in ascending order. So we will have a range of data in ascending order, and we will be having the median of the data. So the median of the data will come inside this box, and the boundaries of this box here will be bound by the lower quartile and the upper quartile. So what are the lower quartile and upper quartile? Lower quartile is the median of the data values between the first value and the median of the total observation. So median between first value and the median of the total population, that will be the lower quartile Q1, median will be Q2, and the upper quartile, which is usually called as Q3, that is the median between your median and the last data point of the total population. So that will be Q3, and this distance or this value between this lower quartile and upper quartile Q3 minus Q1 will constitute your interquartile range, which is called as which is abbreviated to IQR. So now that you know how the box is bound, you need to know how the whiskers are formed. How do you determine the lower extreme and upper extreme of the data? So lower extreme and upper extreme are normally determined by the minimum and maximum acceptable range, which is given by these two formulas here. The minimum acceptable range is given by the formula Q1, the lower quartile, minus 1.5 times the interquartile range. So any data which is outside the lower extreme of the data, which is very below the lower extreme of this data range, will be considered as an outlier. The outlier can be single data points or multiple data points, but if it is lesser than the lower extreme, it will be considered as an outlier. Similarly, the upper extreme will be considered as through the maximum acceptable range, which is the sum of the third quartile, the upper quartile, plus 1.5 times the interquartile range. So any other data point which is higher than the upper extreme, this maximum acceptable range, will form the outlier. So now that we know what the box and whisker plot is and what are outliers is, but what is the purpose of an outlier in a plot? So an outlier just means that it does not, something is wrong with the data. Something outlier just means that it does not belong to the data set. But does it mean that we should be ignoring it completely? The answer is completely not yes. We should not be completely ignoring the outlier. We should be excluding the outlier in terms of further calculations. So if you try to plot a box plot, and then if you find there is an outlier, you should exclude that outlier and try to find out the upper limits and lower limits once again. Then only your data will make sense. But your outlier also has to be presented in a box plot so that you can analyze it later as to what went wrong while measuring that data and the data point in that data set. So this was all that was taught about box and whisker plot. And in the next lectures, we learnt about the propagation of errors. So in the last week's lectures, in week 6's lectures, we saw about how to represent the data. So if we have some x can be represented as x bar plus delta x, where the delta x bar is the mean of the data, mean of x, and delta x is the standard error of the x. So we have x or y. Amit Srivastava. For example, a question being asked for writing the minimum for a given data set, then only minimum value we need to write, or we need to outlier represent. That's the thing. So if they ask the minimum value in the assignment, we will come back. I'll be discussing those problems as well. So if they're asking the minimum value, you should exclude the outlier and write only the minimum value, which is minimum acceptable range which you get. So whatever minimum value you have within the minimum acceptable range, that is only your minimum data value. That's what you should be writing in that answer box. I'll come to that question as well. I will be discussing those questions as well in detail in the later part of this TA session. We'll come back to that. Yes. So yeah. So yeah. Yeah, after box and whisker class, we saw a series of lectures about propagation of errors, and we saw that any data x can be represented as x bar, which is the mean, and plus or minus the delta x, which is the standard error of the data. So how about we have multiple x's and multiple y's, and what if we try to do some functions with them? What if we try to add them or subtract them? So that is what is focused in this propagation of errors. So if we have, let's just say if we have an operation, let's just say z, which involves the addition or subtraction of two different parameters x and y. So how will we represent z in that case? X has x bar plus delta x, and y has y bar plus delta y, plus or minus delta y. So how will we represent z in that case? So the z will also have a z bar, which is the mean of z, and plus or minus the delta z, which is the standard error of z. So z bar is easy to find, which is just the addition or subtracted value of the means of x and y. So z bar will be x bar plus or minus y bar. There is a mistake here. Sorry for that. But how will you calculate the standard error? So the standard error will, in this case, will be calculated like similar to how you add two vectors. Basically, your delta z, the standard error will be the square root of the sum of squares of delta, the standard error of x and standard error of y. So it will be delta x square plus delta y square, the whole root. So that will be your error in z. So having taken z bar and delta z, you will plus or minus both, and then you will present it as the form of z. So we know in the case of addition or subtraction, we can have this. What if in the case of multiplication? In the multiplication, z bar is same. You multiply x bar and y bar, and then you get z bar. In case of delta z, the actual errors will be replaced by percentage errors. So delta z by z will be equal to the sum of square root of sum of squares of the relative errors of x and y. Delta x by x the whole square plus delta y by y the whole square, the whole root. And to find delta z, you take z to this side and then multiply this by z to get the error of z. Yeah. And following that, we also saw how to propagate errors in a function where z is a function of only one variable x. In that case, the z bar, the mean of z, will be the function of the same function at that point of the mean, x bar, and the standard error will be the standard error of x multiplied with the derivative of that function at the mean point. So that will be the standard error of x. And same thing goes for z if it is bound by two different variables. So if z is a function of both x and y, then z bar will be a function of both x bar and y bar. But the standard error delta z will be the sum of squares of the standard errors of each of the variable multiplied by their own derivative at that point. So it will be delta z by delta x the whole square delta x plus dou z by dou y the whole square into delta y square the whole root. And after propagation of errors, there was another short topic on issues in hypothesis testing. So this part of the lecture deals with the various biases and how to properly set up an experiment while we try to prove and disprove hypothesis. And in the earlier lectures and weeks, we saw that while solving a problem, we must come up with many hypotheses and we must frame experiments to negate or disprove all the hypotheses one by one. And the last hypothesis standing will be the true hypothesis for that time. But there are several biases and several other errors which kind of arise due to wrong experimental setup or wrong grouping or so and so. So this is what we try to deal with in this lectures. So first of all, we start with type 1 and type 2 errors in hypothesis. So type 1 and type 2 errors are quite simple. So type 1 error is a error where the null hypothesis is true, but it is rejected by the test. Whatever statistical test we are trying to do to prove the null hypothesis, that rejects the null hypothesis. That's when type 1 error occurs. And type 2 error is where your null hypothesis is actually false, but your statistical test does not reject it. It accepts your null hypothesis. That's when a type 2 error happens. Both of these errors should not happen, and you should prepare your study such that you can kind of avoid these kind of errors. And the one major culprit that is found to cause this errors is something called as confirmation bias. Confirmation bias is something uh, which can be like explained like this. So I'm performing an experiment. I desire any sort of outcome. So let's just say that I am treating a drug A. I am developing a drug A. Let's just say it's some form of weird form of paracetamol. I want my paracetamol to cure malaria. So let's just say I'm developing a sort of paracetamol to cure malaria. So when I am designing the drug and when I am trying to validate the drug, I can, without even my knowledge, or sometimes even with my knowledge, I can design my experiment such that my paracetamol gives maximal output to try to cure the disease, even though it is not. So I am desiring an outcome that my paracetamol drug should work, and I am favoring my experiments. I am favoring my results so that my results will come out positive in favor of whatever desired outcome that I am getting. So this is called as confirmation bias. And whatever experiment we are trying to perform, that should be void, that should not have any sort of confirmation bias. And there are many ways by which you can actually negate confirmation bias. And one of the major techniques is by doing a proper sample of all the test groups and control groups. So what do you mean by proper sampling is you test whatever hypothesis you have on a large number of samples. You do not settle for 10 or 20 or 30 samples. You test your hypothesis on, like, say 500 samples or 1000 samples or increasingly. And another way by which you can do proper sampling is by performing uniform samplings from various groups. And by various groups, we mean that we form experimental and control groups. And even within those experimental and control groups, you form subgroups. And then you try to include as many samples as possible. And then you try to test them in each of them to get statistically significant good outcome. And how can you actually make sure that you can do uniform sampling in experimental and control group? There are two different methodologies. One is a completely randomized design. So you just randomly take any group in any sample and place it in any group of your choice. That is completely randomized design. And then another way is randomized block design. So you take any group or any subgroup and then you randomize all the samples in it so that you don't know which one is kind of susceptible to your hypothesis or which one is non-susceptible to your hypothesis. And in the last part of the lectures, we see about single blind and double blind studies, where we see what's the importance of single blind and double blind studies. So basically, single blind and double blind studies are performed to eliminate any sort of bias, confirmation bias, either from the experimental side or from the participants' side or from the patient's side. So let's just say that let's just take the same example. I want my drug, I am having a drug, I want my drug to work on these people. So I can purposefully take more healthy people toward my experimental side. And even though the drug doesn't work, I can tell that the drug works and people are healthy, so my drug is working. And then I can purposefully push the non-healthy people, less non-healthy people towards the, let's just say, control side, and then say that without my drug these people aren't like recovering, so we need a drug to proceed forward. So these kind of biases should be eliminated, and that's why a single blind study is conducted. A single blind study is where the researcher doesn't know the treatment groups. So for example, I am the main investigator of the study, of any study, for the drug study, and I should not be involved in any other study. So instead, what I should do is I will know which, let's just say I have like 10 vials, and I know that these 5 are the actual drug, and these 5 are something called as the placebo. Placebo is nothing but, placebo is any substance which feels, or like, it acts like the drug, but it's not the drug. So it won't do anything to your body. So that's the placebo. So I know which drug is my which vials are my actual drug and which vials are the placebo. But I will not be experiment. I will not be trying the vials on the patients myself. So instead, what I'll do is I'll ask a middleman who doesn't know which one is the experiment, who doesn't know which one is the vial or the which one is the drug or the placebo, and ask him to inject it to my experimental and control groups, such that neither me, I don't know who the middleman is injecting to, and the middleman doesn't know who, which one he is injecting to which kind of person. So that's called a single blind study. Double blind study, on the other hand, is performed when a participant, let's just say, a participant also doesn't know what he's being injected with, whether he's being injected with a drug or a placebo. So this is to also eliminate any kind of placebo effect from a uh, eliminate sort of a placebo effect from the participant. The placebo effect is something where um, a person takes placebo, but then he thinks that he has actually taken the drug, and then something happens in his body which is positive to our outcome, but is performed only due to the placebo. And single blind studies and double blind studies are often, like, in summarizing this, single blind studies and double blind studies are performed to eliminate biases between the participant as well as the experimenter. So these were the concepts which were majorly discussed in the week 7 as well as last parts of week 6. So I'll open the room for a small doubt session with respect to the concepts in till like 3:30. After that, I'll start discussing the answers for week 6 and subsequently week 7.
What is the significance of error bar, that is viscous bar, outlier in the viscous bar?
Ah, so so that's what. So outlier is something which is either an outlier data point is something which is not bound to the minimum and maximum acceptable range in a box and whisker plot. So any outlier must be presented in a box and whisker plot. I mean, not just because it's not bounding to the range doesn't mean that it should be negated out of the box and whisker plot. It should be included such that it can be studied later. The data point can be taken out and studied later as to what went wrong with the data set. And another significance of the outlier is that you can, when you negate the outlier and recalculate it again, sometimes, not all the time, sometimes the box and whisker plot changes again, changes its characteristics again, which will help you to study the overall population better. And that is what I think Amit Srivastava also said. For any data, given data asking to write Q1, Q2, Q3, median and median, is there any need to calculate outliers? That, like, you need to calculate outliers, but like you need to find out the outliers, but you can't take those outliers into calculation. Yeah. And in these questions, I think the questions of week 7, I don't think your outliers influence your box and whisker plot that much.
Okay.
Yes.
So when we calculate the mean, like it's just usually how we calculate it. We haven't done any particular way of doing the mean, right?
Yes, yes. Mean is just like sum of observations by divided by the total number of observations. That is, like, we haven't done any different stuff. But in box and whisker plot, like if you're talking about box and whisker plot, then box and whisker plot doesn't involve the mean. It instead involves the median. Yeah, the median is the middlemost observation in a set of data points.
So the first set of questions, like they have asked the mean also. So it's just
Yeah. There was
observation.
a mean. We'll come back to that. Yeah, we'll come back to that. So I think its time is already 3:30, and I think there are a lot of mathematical questions to be discussed. So I'll straight away jump into the question and answers, and if we have time in the end, we will have uh, we'll allocate time for doubt session separately. Yes. So yeah, so and week 6 and week 7 almost involved questions which involve the use of Z tables and T tables. So I think someone shared Z tables to the Google Groups discussion before I did. Many thanks for that. But I have also attached the Z table and T table in this PowerPoint presentation if anyone wanted to access it later. Yes. So with that, I'll jump straight away to the problem solving assessments. So first question, like for this is first I'll discuss the week 6 questions, and later we'll go on to week 7's questions. So first question, suppose you have measured the concentration of a substance in a solution by taking 25 samples. So they have the concentration in ppm of 25 different data points, and they are asking if you are to report the result in a paper, how will you specify the measured value around to 4 places. So if you have to measure the value, then we have to report the mean and the standard error. So that's what they have asked in the first question. So So this mean, we should ignore the outlier and simply write the minimum. Yeah, we should actually ignore the outlier for calculation of the minimum mean. I'll come back to that later. Yeah, answer is 1.0596. So if you add up all the data points here, your sum will come to like 37.74, and we have 25 observations. So mean will be like sum of all the observations, 37.74, divided by 25, which will be 1.5096. Yeah, so there is a like, even if you like, I know that all the people won't be finding the mean through the same method. Some of you may be using calculators, Python, Excel, and all those stuff. So and different compilers will give you different answers. So NPTEL gives you a range of values within which if you write your answer, it will be correct. Only thing is that you have to round off your answer to 4 decimal places. So actual answer is 1.5096, but it is correct if you write your answer anywhere between 1.5095 and 1.5097. Yes. And second question is error bar. Yeah, I think last week also many people got confused with this. By error bar, they actually asked the standard error and not the standard deviation. Yeah. So standard error, you know that standard error is given by the formula sigma by root of n, where sigma is the standard deviation of the 25 samples, and standard deviation you can find out that it is 0.0547, and if you divide that by root 5, you will get the answer as 0.0109. Yes, and yeah, like once again, many people are saying 0.107. Okay, Piyush is saying 0.107. I think I don't know if you mean 0.0107, then that would be that would also be a correct answer.
0.01.
Yeah, then that's a correct answer. So the acceptable range is 0.0106 and 0.0110. As Gupta, you also told 1.0107, but if it is 0.0107, it's correct. I don't know if you have typed it wrong. Yes, but this is how you would actually calculate the okay, if it was right, then it's okay. But this is how you would calculate the error bar. So you find the standard error, which is the standard deviation divided by the root of number of samples. Yes. And third question was, define a range in which the true value of the concentration must lie with 95% probability. So we know that 95% probability is like 1.96 times the standard error of the mean. So we have a normal distribution. Yes, Gupta. So I mean, you have written 1.0107 instead of 0.0107. That's why I was a bit doubtful. Yeah. So in this question, we'll come back to this question. So we know that this if they ask by 95% probability or 95% confidence, it means that your area is covered till the 1.96 times the standard error of the mean. And 1.96 times the standard error is, so we know the standard error from previous question. So here we know the standard error is 0.01909. So we'll multiply that by 1.96 and we'll get this 0.0214 value. And then what we do is we find the x range. So x range is the lower and upper limit from this mean value, and then you will add and subtract. Sorry. Okay, these are two different questions, right? So they have told us the range is x to y. So x is the lower limit and y is the higher limit from x from the mean. So for calculating x, you must subtract the 1.96 standard deviation from the mean, which will give you around 1.488. Yeah, 1.49 also works. The acceptable range is between 1.48 and 1.49. And similarly, y range is given by adding 1.96 standard error to the mean, which will give you 1.531. And the acceptable range is between 1.53 and 1.531. Yeah, I hope everyone is clear with the questions. So I'll go to the next question. With what confidence level can you state that the actual value lies in the range 1.5 to 1.5192 ppm? So I hope everyone gets the question by what they are saying. So we already have the mean 1.5096, and they're given 1.5 and then 1.5192, which is like 0.0096 of ppm of both the sides of the mean. So yeah, so how we should try to solve this single question is, we want to find the confidence level. So the confidence level is nothing but using the Z table, you will find the Z score of according to the Z score, you will find the area covered, and then from the area covered you will find the probability or the confidence level. So you have the mean 1.5096, and then from 1.5096 you take the lower limit 1.5 and subtract it to get the 0.0096 value. And to find the Z score, you divide this difference by the standard error 0.010947 to get the Z score of 0.8769. And if you go to the Z table, like I showed you the Z table already, 0.8769, you get an area of 0.8078. And to find the actual confidence, so 0.8078 is to the left of this whatever Z score we have here. So left of 0.8769 standard deviation to the mean, the area is 0.8078. So to find out the confidence level, what you do is you subtract that from 0.5 and then multiply it by 2 to get 61.56, 0.6156 or 61.56 percentage. And the NPTEL accepts any value if you want, whether you write it in percentage or in decimal places, it doesn't matter, but the answer should be within 61 to 63 percentage or 0.61 to 0.63 confidence level. I hope the answer is clear. Okay, I'll go on to the next question then. Scientists could take only 10 measurements on the molecular weight of a new compound. So they are given 10 measurements and 10 different values. They are asking us what is the standard error in mean. So same thing. So you find the mean of all the 10 measurements values, and then you find the standard deviation. So you know the standard error is standard deviation divided by the number of samples. Here n is equal to 10. So you divide the standard deviation 2.73 divided by the root of 10, and you get 0.8649. So that will be the standard error. And the acceptable range for any answer is 0.84 to 0.88. Depending on whatever compiler you use or whatever calculator you use, any answer from 0.84 to 0.88 will give you an answer. But if it comes 1.82, 0.82, Okay. Can you explain it to me, Gupta? 86 is good. Sonu says 86. 86 is good. Like 0.86, that would be like 86 is good, but 82 will not be a correct answer. Like 84 to 88 will be a correct answer. 0.82, okay. Yeah, Sonu 0.86 is correct. Avani and Gupta, okay. Like, did you double check or cross check with any other thing, calculator or any other Excel or anything else? Like, maybe there was any like typing mistake. I have done via Python. Okay. Yeah, Sonu Excel works actually. Like many problems, I think you can use Excel to get the standard error. You might not have formula, I guess, but I think you will have formula in Excel for standard deviation. If you do that and divide that by square root of 10, I think you'll get the answer. Gupta and Avani, can you tell me what standard deviation did you get? Because I think root of 10 is like common, like no one can make any mistake in it. Like root of n is equal to root of 10, that no one can make any mistake. Prashant, 2.7351, how you got? 2.7351 is the standard deviation, I calculated it. Vinita, 0.8655 is correct only. 2.5947. So Gupta is also saying 2.594. Yeah, Vinita, you are correct. But Heba, Avani, and Gupta, I'm a bit confused how you got 2.5, 2.5, and all those things. Okay, standard deviation calculation. Maybe I can just open you guys Excel and then like show, like we can just do a live demo of this thing in Excel and then we can find. Yeah, 2.735142 is correct only, I think. Okay. We'll come back to this doubt later because I think many people are also waiting for session 7 answers. So uh, we'll finish this and we'll come back to this question later. Yeah. Um, do the data provide sufficient evidence to say that the compound's molecular weight is less than 46.5? Here, sufficient evidence implies that the probability that the statement is wrong is less than 0.01 or 1%. So they are just asked. So we already know the mean of the compound from the previous questions, and then they are saying that if whether the molecular weight will be less than 46.5 with a higher confidence interval. And since we have 10 samples here, we can't use the Z distribution. Instead, we have to use the T distribution. So we have the mean 45.31, and then for the t distribution we have to use the degrees of freedom 10 minus 1, which is 10 minus 1 is which is equal to 9, and then we have to find the z score, in this case the t score. For that we have to subtract the mean value from the expected value of 46.5, and then divide it by the standard deviation, standard error, which is 0.8649, and we will get this z score 1.379. And if you check the t table for uh, so we have this t value here, and then if you check the t table for the t value for degrees of freedom at 9, 10 minus 1, and at 0.01 confidence, because we want confidence of 0.01, so there we'll get a t value of 3.25. So obviously this 3.25, it is within this, whatever z value we have, it's within 3.25. And so whatever molecular weight we are proposing it to be 46.5, it will not suffice, it will be greater than 46.5. Therefore your answer will be no. Do you okay, 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 then I'll go on to the next question. So suppose you have detected a systematic error in your experiment, what steps would you take to eliminate it? So this is a multiple select answer question, and options are central limit theorem, two or more different techniques, larger number of data points, and calibration of systems you're using. Gupta is saying B and D. Yes, that will be the correct answer. So to verify that if you have made an error or something, you need to make the same measurement using two or more different techniques to check that whether you're getting the same value across all the techniques. And the other one is to make the calibrate the instrument that you're using to measure the quantity. And ninth question, an error bar around the measure value implies it's a single MCQ question. Only 1 point would be correct. C. Gupta is saying C, correct answer. The probability that the actual value lies within the error bar is 0.683. That is one standard deviation. And last question, standard error of the mean is, it's a multiple select question, and Gupta once again is B and C is correct. So readings obtained divided by the square root of number of data points, and then standard deviation of the distribution of the mean values according to the central limit theorem. And the last question, I think we discussed this last week as well. The bioscientist can take 6 observations from his measurement. How can one define the confidence level? So since it is less than 25, he can't use the Z distribution. Instead, he has to go for the T distribution table, which will allow for samples less than 25. Yes. So yeah, Sonu, you're also like C was the correct option. Yeah. So I think we have that standard error question to be discussed yet. What we can maybe do is we can uh, go over the week 7 questions. Like Gupta, you can go ahead. Yeah.
Sir, there is a doubt regarding the number of observation in the sample. So if it is 25, we should go for T distribution or Z distribution.
25 or above, you can go for Z distribution because you're
Means up to 24 T distribution,
yeah,
and to 24 to more Z distribution.
Z distribution. Actually, there isn't any clear bifurcation between Z and T, because if you see some T tables, uh, even for degrees of freedom till 30, you will have some values. Yes, but for values greater than 25, you can go for Z distribution without any hesitation. That's the hunch.
No, means greater than 25 or 25 and greater than 25.
This was what 25 and greater than 25, you can go for Z
Okay,
distribution.
both the things. Okay,
Exactly. Yeah.
right, sir. Thank you, sir. Thank you.
Okay. Okay. Um, okay, we'll go for the this thing week 7's questions. I have almost curated like 7 questions. Like So I'm not allowed to discuss all the questions per se. So I have sort of taken out all the example questions, like any questions from each question type, just to say how each problem can be proceeded with. So we'll go ahead with this. Prashant, you can go ahead. Yeah, you can go ahead.
Yeah, sir. Actually, standard deviation calculation because during the exam the scientific calculator would be there. I
Yeah,
think MS Excel not available. So please explain me once the standard deviation calculation how to do.
sure,
Second,
sure.
if possible, please during the section during the sir lecture classes also, sir one to two time one time he explained. So later on, just he taken the value only. So the Z value and T value, how to take from the Excel. You also forwarded last week. The
Yes,
table you
yes.
provided. So just one more time, please remind that T value how to take or Z value how to take from the values.
Sure, sure, sure. After discussing this week 7 questions, we will go back to week 6, that standard deviation problem, and we will go. I'll also tell you how to access the Z table and T table.
Thank you. Thank you. Thank
Yeah,
you.
yeah, sure. So we'll start with week seven's questions. So suppose there are two strains in a species of bacteria, one benign and another virulent. So you have read a paper that says more than 32% of the bacteria are virulent, and you have repeated the experiment and cultured the bacteria. Then you pick up a bit of the culture and find 200 bacteria in it. On closer inspection, you find that 76 of them are of the virulent strain. Does your observation support the report in the paper with a 95% level of confidence? So 76 out of 200 in your experiment is virulent, but in the paper 32% are virulent. So can you accept the can you accept or support the paper with a 95% level of confidence? That's what you need to find. And we'll use them, like for this problem we'll be using the measurement of proportions concepts. So from the paper you know that 32% bacteria are virulent, so your p will be 0.32 and 1 minus p will be 1 minus 0.32, which is 0.68. And your your experiment 76 of 200 bacteria are virulent, so that ratio, that measured proportion, is 0.38. So what you will do now is you will frame an null hypothesis that your p-value is less than or equal to 0.32. If you if you are able to successfully reject your null hypothesis, you can accept an alternate hypothesis that your p-value is greater than 0.32, which will be your 0.38. And first, before going to the experiment, you need to find out whether your uh, values, whether your p values will give a normal value. For that, you need to multiply p and 1 minus p with the number of samples, which is 200. So your 0.32 and 0.68 will be multiplied by 200, and both of them are 10. So you are sure that your p cap distribution, the measured proportion distribution, will be normal. So after that, what you do is you calculate the value of standard deviation, which is given by this formula. So you have p, you have 1 minus p, you have multiply them, divide them by n is like 200, and then you take a square root of them. So it will give you this value 0.03298. And then what you do is you have the standard deviation. So you calculate the z score using your mean p value and then your ratio, measured proportion, and then you get a z score of 1.82. So use the z score in the z table, and you find the area of the z value of 1.2, which is like 0.9656. So for the z score of 1.82 standard deviations to the mean of 0.32, you have an area of 0.9656. So how do you calculate the probability from that? You subtract 0.5 from this value and multiply it by 2 to get a probability of 93.12%. So 93.12% is obviously less than 95% of your level of confidence. So you can't like you have to accept your null hypothesis, and you have to say that you can say with confidence that your observation supports the report, which has with the 95% level of confidence, because your data point with 0.38 lies within the 95% value. So your answer will be yes for this question. Yes, yes, yes. Kangembam Biren Singh is saying Z is less than 1.96 also suggests the answer. Actually, very, very true. That was actually a shortcut method I told everyone in the call last week as well. Because 1.96 is like 95%, 1.96 standard deviation says 95% level of confidence. Your Z score is already less than 1.96. So you can say already from here itself you can say that with confidence that you can support your observation. Yes. So we'll go on to the next question. Same question, like we already had calculated the Z score for this as well, 1.82. So the next question's answer will be what is the value of Z? So Z score will be 1.82. Gupta, you can go ahead. Sorry.
Yes, sir. I have taken in this questions. The approach is 1.65, which is less than 1.82. So it means we are in the rejection region. So I have rejected the null hypothesis. On the basis of that, I am saying I am supporting this
Yeah, you can also
report
you can also go by that method as well, but you might want to be careful with that method as like some cases that may not work.
because I have taken the right tail test, sir.
Yeah. So you can consider you can go with that method as well.
Okay, okay. Thank you, sir. Thank you.
Yeah. Amit Srivastava, like, can you give an example? Because I don't think you need to write any range in a box. You just need to give a value. Whatever range I'm giving, that is the acceptable range for NPTEL because not many people will be giving the same answer. So, a bit more to the left of right of the actual answer is also accepted. That's why the answer has a range. And second question, we already calculated the Z score in the previous problem itself. So Z score is 1.82 and anywhere between 1.81 and 1.82, the answer should be fine. So I think, yeah, this all is the same from questions 3 to 8. I think you have the same type of questions. So 1.8192, I think, I don't, for what question is this, Vinita Adhikari? Yeah, Z score, yeah. Yeah, 1.8192 is fine only. It falls between 1.81 to 1.82, so it should be fine. Yeah. So question number 3 for the minimum, find the minimum median, that is Q2, lower quartile, upper quartile, and the maximum for the following data obtained in experiment. Identify if there is any outlier and draw a box and whisker plot. So before going to solve the next 5 6 questions, you need to, like, it's a wise method to actually find all the parameters and try to get the whisker plot parameters. So the first way to do it is by arranging all the numbers in ascending order, which I have done here, 19 to 88. 19 will be the minimum value, 88 will be the maximum value in the uh, ascending order. Your median will be the middlemost plot. So here we have 15 values. So median will be the eighth value from the start, which is 73, and Q1 will be the median value from the first to the eighth, which will be the first to the seventh, which will be the fourth value, which will be 56, and then Q3 will be the median between the ninth and the 15th, which will be the 80. That will be the median of that will be Q1, Q2, Q3, and the interquartile range is Q3 minus Q1, which will give you 24. So we have these values. Now we have to calculate the minimum and maximum acceptable range. So the minimum acceptable range, we already saw a formula. It will be Q1 minus 1.5 times the interquartile range. So 1.5 times the interquartile range will give you 36, and Q1 is 56. So 56 minus 36 will give you 20. So 20 is the minimum acceptable range, and the maximum acceptable range is Q3 plus 1.5 times interquartile range, which will give you 80 plus 36, which is 116. So if you take these values and compare this in all the points, you can find out that 19 is it's outside the minimum acceptable range of 20. So your 19 will be outlier, and if you remove 19, if you remove the outlier, the next most minimum value is 24. So your minimum value will be 24. That will be the answer. And next, like all the successive questions, we have already calculated all the answers. So your Q1 will be 56. Fifth question, Q2 will be your median, which is 73. Q3 will be your 80. Q3 here. Your maximum will be your maximum, as in they are asking the maximum value in the point, not the maximum range. So you should give your maximum point, which is like 88. So your maximum will be 88. And mean you have to find out here. Mean you have to find out by so for mean, what you have to do is you have to remove 19 because 19 is your outlier. You can't take your outlier for all the data calculations, but you have to just present it in the plot. So you take all the sum of the values from 24 to 88 and then divide that by 14, because if you remove the outlier, there are only 14 observations left. So you will get the mean as 69.28571, so on and so forth. Your acceptable range will be between 69.28 and 69.29. Is everyone clear with this set of 5-6 questions? 3, 4, 5, 6, 7, 8, I think. Yeah, no one has any doubts, right? Like I'll just wait for 1-2 minutes before going on to the next question. Dakshata, I can go ahead.
So one more time, can you explain?
The left side concepts.
Last one, Q3 maximum.
Okay, okay. Like which ones are you unclear with?
The
Like,
maximum value, Q3
oh,
or maximum.
oh, okay, 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 okay. So you have this. So we have the question, they have given this numbers. So to find the mean, median, and all those stuff, what I have done is I have arranged them in ascending order. So minimum value is 19, maximum value is 88 in this range, and then I have arranged them in ascending order. So you have to find medians. Medians and mean are different. If you are not familiar with statistics, median is the middlemost point in the entire range. So you have 15 numbers here. So in 15 numbers, which is the middlemost value, the eighth value? So 1, 2, 3, 4, 5, 6, 7, 8. 73 will be your eighth most value. So that will be your median. Median you assign it as Q2. Q1, which is the first quartile, that will be the median from the 0th value to the 7th value, the value before your median. So you have 7 values here. So middle of 7 is 4. So the 4th value will be your Q1. So 1, 2, 3, 4, 56. So 56 will be your Q1. Q3 is similar to Q1, but it's on the other side of the median. So median to the last point. So from here you have again 7 observations, and the 4th one is the middlemost observation. So 1, 2, 3, 4, 80. So that will be your Q3. So we have Q1, Q2, Q3. Now we have to find the range. For range, we have this formula for minimum value of range and maximum value of the range. Even in the lectures and even in the recap, I have given a formula that minimum range is Q1 minus your 1.5 times interquartile range. That will be your minimum range. Kangembam Biren Singh is saying, should not the minimum of the data remain the minimum? We remove outliers for further calculation. So that's what. So outliers are just presented. They are not included in the box and whisker plot. Do you get it? So the plot ends at the minimum range. The whisker, the last whisker, the plot ends there. Below that, if the outlier is there, those are just there. Like you can just visualize it. You can't use it for any sort of calculation. And same goes for any data point above the maximum range as well. Sir, I have taken Panda series and taken summary of the disease data. It has given the values like 58, 73, 79. So one major thing here is that one thing is that in the main exam, in the final exam, you can't use pandas and Python and all those stuff, Excel and all those stuff. So I would advise you to do the calculation by hand itself or using any normal calculator. Another thing is that different people have different methods of calculating the mean. So uh, how Python might have took it is that it would have taken mean from 19 to 73. One second, Dashara, I'm coming later to your doubt as well. So by pandas, I think it would have taken values from 19 to 73. They would have included 73 as well for the calculation of Q1 and Q3. So I think if you include 73 as well, you will get the Q1 as 56 plus 60 divided by 2, which is 58 average. And then Q3 will be your 78 plus 80 divided by 2, which is 79. Yes, but you can't. So for for a very simplified version of the box plot, what we are doing is we are taking numbers which are on the range. We are for Q1 and Q3, we are taking numbers which are on the range itself. We are not going for any addition multiplication stuff. So for further any calculations, I would suggest you to do it by hand as well and see if you are getting the same answer as well. Dakshata, you can go ahead with your talk.
Sir, my mean value is 65.93.
Your mean is 65.93. Did you
Yes.
take 19 also into account?
What?
While calculating the mean, you will add all the numbers, right? Did you add
Yes.
19 as well?
Yeah.
Yeah, I think if you add 19 as well, you will get a lower mean. So 19, we have found that 19 is an outlier already using the minimum and maximum acceptable range. So you remove
So
19. So you remove
okay.
19, start adding from 24 to 88, divide that by 14 because now excluding the outlier, we have only 14 observations.
Okay. Okay. Okay.
Yeah.
Understand, sir.
You
Thank you.
can put it in the chat box if you're getting the same mean as well. Piyush, you can go ahead.
If the values are odd, even number, then how to calculate median?
Sorry?
Here, 15 values are given. Therefore,
Yeah.
we have calculated middle value as a median.
Yeah.
But if values are given odd number, suppose 14 values are given,
14, if
then
14,
how?
14 values are given, then you take the average of the 7th and 8th value. Yeah, I think this point was discussed in the lectures as well. So median of odd values is the middlemost value. 15 values are there, then you take the 8th value, which is the middlemost value. 14 values are there, you have 7 and 8 are middle values, right? So you take the average 7 plus 8, 7th value plus 8th value divided by 2. That will be your median.
Okay.
Yes. So I hope everyone is comfortable with the concepts of Q1, Q3 and interquartile range and mean. So we'll go on to the next question, which I think is deals with the propagation of errors. So no 67.64, I don't get it like median. I think it's a 70 only, like because there are only 15 observations. Okay, so next set of questions will focus on this propagation of errors. We have conducted an experiment to measure two values x and y. So they are given x values and y values, and for each values they are given mean, standard deviation, and number of data points, n value. And I think, if you, I think, okay, these are like part of the questions out of which if you get the answers, you can answer all the rest of the questions. So ninth question is basically x plus y. You add x and y. What will be the value and plus or minus the error? So a value you just add 4. So it will be 4.8 plus 6.6. It will give you 11.4. So 11.4 will be the answer for this. I hope everyone is clear. So if you have two values, uh, any value will give you x bar. Mean x bar means the mean plus or minus the standard deviation or standard error actually. So mean plus or minus standard error. So here they are given standard deviation. So for a you have to just give the mean of the addition. So you just have to add the means of x and y, which will give you 11.4. Second question, x and y, same question, a plus or minus b, but b value. So here B value just means standard error, not standard deviation. So if you just do the, so we discussed the standard error of the sum will be like square root of sum of squares. But here what you have to do is you have to take the standard error first. You have to how do you get the standard error? You already have the standard deviations 0.36 and 0.28. You divide that by the square root of n. That's how you get the standard error. So square root of 36 is 6 and square root of 49 is 7. So you have got the standard errors, square both of them, and then take a whole square root of them to get 0.07211. And I think it would be acceptable if any answer within 0.07 and 0.08 are got. So next question is instead of x plus y, they are given xy. Xy is equal to a plus or minus b. Here they have asked a value. So if you have multiplication, you can, for the a value, which is the z bar, you can just multiply 4.8 into 6.6. So the answer will be 31.668, and your range will be between 31.6 and 31.7. So any answer within this will be accepted. And I think this next question is also b value. So b value, we have already discussed this in the propagation of errors. So if you are multiplying something, you don't take the absolute actual errors, you take the percentage errors or the relative errors. So your delta z by z will be equal to the square root of delta x by x the whole square plus delta y by y the whole square. And so you take z to the other side and then calculate b value. So I think that's all the questions I have for this question.
Sir,
Yeah.
the 12th question, can you explain one more time? The delta is not understand.
Yeah, so in 12th question, so once again, I'll just go back to the, yeah. So you have to use this formula, which is just given here. So they have asked z and they have given x into y. They have multiplied x into y, and then they are asking us a value and b value. A value is simple, you just have to multiply the means of both the questions. And for the b value, what you have to do is you have to use this formula. So delta z will be your b value, and you already know the z value, which is the x into y, x of mean of x into mean of y, that will be your z value. And then you have to square root it, and delta x you already know, delta x you already know, x already you know, delta y you already know, y bar also you already know. So what you do is you transpose z to this side. So delta z will be z into the whole root of this equation.
Which value is delta z?
Delta z you have to calculate. That delta z is the answer.
Okay. Z value is mean and delta x is, and delta y we calculate.
Yes.
Then divided by the x and y values.
Yes, x and y is just x bar and y bar. You just substitute x and y with x bar and y bar. And delta x and delta y you've already calculated. They have already given you the standard errors. You have to calculate the standard errors, right? So you just substitute the standard error in them, and then you proceed with that. So it will be that 0.36 divided by 6 will be your standard error for this one, and 0.28 divided by 7 will be the standard error for delta y. I hope it's clear.
Arjun Mayuresh here. Can I ask one question?
Yeah, sure, go ahead.
Yeah. Actually, no. This there is you would have explained for sure, but still once more I want to ask. This is
Yes.
regarding box and whisker plot. No.
Yeah.
In that box and whisker plot, it is asked to report minimum and maximum values. So now I am able to do this entire sum, but only thing is, no. Suppose we are getting some extreme values, extreme minimum and extreme maximum,
Yeah.
and suppose data lies within that. The given data is lying. For example, it is given as minimum value as 19, 19. Okay, in this
Yes.
data set.
Yeah.
And I'm getting something like, let us say, 25. So I
Okay.
should report 25, or I should report.
You should report 25 only. If 25 lies within your range, like minimum acceptable range, and 19 does
Yeah,
not, then you should report
yeah.
25 only.
So here in the data set given,
Because
after
19.
after 19 there is 24 in the given data set,
Yeah, so you
and
should report.
I'm getting minimum as 20. That means 24 I should report in that case.
Yes, yes, yes. Because
No, my question is whether I need to report 20, which is what I'm getting from
no, 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 no.
Q1
So
minus
they haven't asked the
Q1 minus that.
yeah, yeah, they haven't asked the minimum acceptable range, right? So in that case, you can report 20. They have asked for the minimum value in the data set, which is excluding the outlier.
No, because no, I got confused because question says that what is the value you can report. So it should be reported only from data set, not from the calculation.
Yeah, it should be reported from the data set, but excluding your outlier.
Okay. Okay, right.
Yes.
So clear, clear.
Yeah. Damodaran Maniranjan is saying how to calculate ln-x. You can take your normal like scientific you can use your calculator in your this thing as well. Like there is no any formula. Like you have to use your natural logarithm book. Like, if you want to calculate naturally, you would have to use the natural logarithm book, which I don't think it will be provided in the exam, nor I don't think many people will have. So what you can do is you can use your scientific calculator. Just you'll have that ln option there in the normal calculator. You can just put ln of whatever value you want to have.
So for x, we'll be putting the mean of x that value, right? For ln.
Yeah, yeah, yeah. Vinita Adhikari is saying question number 13, 15. I don't have the questions with me right now for 13 or 15. Let me see if I can get them somewhere. Questions 13 and 15. I think Vinita, I think you did log instead of ln. I think you might want to divide that by your 2.3, which is you have to convert that into natural log, I think. Yes, yes. So sorry, question number 12. Question number 12 also, I don't think you would be getting that much answer because it's the percentage error, so it will be coming less than 1 only. Like put some thought into it. But I think the answer
Sir, question number 12 answer is 0.10.
is 0.10. Yeah, that can be an answer as well. Like I don't know if you can say that is the right answer as well.
So for 14th one, for b value of ln(x), we should do delta x by x, right?
Once again, I just see what's the 14th question. 14th question, ln x a cross. So ln x a plus b, b value is yeah, yeah, you should do delta x by x only. Yeah, you should do the same thing as multiplication, delta x by x into delta y by y. Up to that.
Mayuresh here. So this A plus minus B is what is A and what is B is a limit tolerance band or error
So
band, is it not?
b is the error. So if they ask
Okay, I
a,
mistook it like A is a upper band and B is a lower. A is a upper maximum and B is a
no, no,
lower lower limit.
A is the mean and b is the
Yeah.
error value.
Oh, okay. That's a standard error.
Standard,
One
yeah,
sigma, right?
yeah, standard error 1 sigma, exactly.
Okay.
It won't be 1 sigma per se, it will be 1 sigma divided by root of n, because
Yeah, yeah,
sigma in
correct.
this context is standard deviation.
Yeah.
I'm getting a lot of interference. Can you people mute your mic? Yeah, thank you.
12th answer is 0.10. Is that correct?
answer is I think it's around 0.1, 0.3 only.
0 point.
I think it's around 0.1, 0.10 only.
It's 0.10 only. No?
Mm, 1 second, I would have to work that out because it's XY, right? 1 second. I don't think so. I think it will be around 0.4 or 0.5. One thing is that did you try to divide the answer by standard? Like, did you use standard error or standard deviation to find the thing that would maybe, or maybe you wouldn't have multiplied it by the uh, z value.
Yes.
I guess it is
Multiplied by z value.
0.44. Yeah, yeah.
Yeah, I think 0.44 would make, yeah, that would
It
be
is
correct, I guess.
0.44.
Yeah, yeah, yeah.
0.44. I'm getting, yeah.
I think you would want to multiply that by z value. Yes, because delta z by z would be I think around 0.1. Z value, your Z value would be around 31.16, like we discussed before.
I can get the answer, sir.
Sorry, you can't get the answer.
Yes.
So you got 0.1, right? Dakshata, can you hear me?
Yes, sir.
Yeah, so you got 0.1 as the answer, right?
Yes.
So we I think what mistake you might have done is you might have taken delta z by z as 0.1 as the answer. So what you can do is you can multiply 0.1 into z to get delta z.
So z value will be 11.4 now.
Z value will be 4.8 into 6.6, which will be like 31.16, I think. No,
So
not
both are
the,
the addition.
no, both are multiplication. It's xy. Xy is equal to a plus or minus
Okay.
b for
Okay.
the
Okay.
question.
1 second, sir.
Sonu is saying upper limit is equal to mean plus standard error. No, no, no. Upper limit is median. Sorry, upper limit is third quartile Q3 plus 1.5 times of um, interquartile range, and then lower limit is Q1 minus 1.5 times of interquartile range. I can maybe put it here.
Sure. How can we find it in this question? Like the upper limit and lower limit.
No, 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 no. Sonu, I think he is asking for another question altogether. Sonu, I think he is addressing the box plot questions.
Upper limit and lower limit is asked only for this, I think. Uh, that the other one is the
Arjun
maximum
Mayuresh
and
here.
minimum.
Probably same doubt I'm also having. Can I speak?
Yeah, 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 sure, sure, sure.
Hello. Yeah, probably this question number 15 and 16, it is asked to report the lower and upper limit. And the premise given is it should have a 99% probability. It is given in the information in the question. So with 99% probability, do we need to take 2.58 times of the standard error then? So whatever mean value is there plus minus standard error into 2.58. So that will be the lower and upper limit.
Once again, Mayuresh, I'll get back to you.
Yeah,
Can you
yeah.
repeat the question once again?
See, question number 15 and 16, it is asked to report lower and upper limits
Okay.
of this data. And I assume this data is for the last, that is for
Yeah, yeah,
lnx.
LNX.
Yeah, lnx only. So for that, it is asked with a 99% probability.
Okay.
That I don't know whether it is correct because it is asked
99,
in the start of the.
99% means you will take 2.58 standard errors.
No, but I'm just trying to connect the question, which is like the premise is given much earlier. Like when before question number 9, there were a lot of data given. So in that only it is given 99% probability. So when it is asked to find out lower and upper bounds, do we need to take for 99%
Yeah. 99%.
probability?
99%. 99%. That's the. So you have to take lower limit with a 99%, lower limit of ln of x with a 99% accuracy.
Yeah, the question is very confusing. Would you mind like how to work it out?
Probably it is a mean value plus minus 2.58 times of the standard error.
Yes, exactly.
That will be giving the lower and upper limits.
Yes, exactly.
Okay.
So you already have, one second, I'll go
Yeah,
back. Where is that?
99% is 2.58, no?
Yeah. So this is the question we have. Main question is x value is mean is 4.8, and then they are asking us to find ln of x. They are asking to ask us to find ln of x, and for that ln of x, I think they are asking us to find lower and upper limit.
Yeah. Only thing is whether is it related to ln of x? That only is a
Ln
question
of x
now.
or x. Okay.
No, because that is question
Okay.
number 13, 14. Whether is it connected to 15, 16? That's the
Guys,
question.
guys, 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 guys. No. I don't think it's connected to lawn. So wait.
Okay.
Yeah, let me just go over the questions. Calculated the estimate values of the quantities x plus y, xy, and ln x, including the error bars. Define a range of x in which the true value of x must lie with a
It is
99%
only for
probability.
x.
Yeah. Sorry, guys. Like very sorry. Like I think I misunderstood the question. So I also type it down in the question as well. So questions 15 and 16, uh, consider only x, do not consider ln of x. Yes, so you already know the mean of x, which is like 4.8. You have to plus and minus 2.56 standard deviation, standard error from that, which will give you, I think, 2.56 into 0.06 some values, and you have to add and subtract from that to get value, and then you can put that as lower and upper limit. This is for questions 14 and 15 and 16. So please repeat. So okay, so they have basically what they have asked us to report the lower and upper limits of x with 99% probability confidence interval. So I'll also type it in the chat box. 99% confidence interval. That would mean that you are covering 2.56 standard deviations of. Okay, wait, I think my mic is off. Sorry, I think my mic went off. So yeah, 99% confidence interval means that you are covering around 2.56 standard deviations of the mean. So you already have the mean and you already have the standard deviation, which is like standard deviation which is 0.6. So your standard error will be 0.36 divided by the root of 36, which is the number of observations, which will be equal to 0.06. Yeah, so 2.56 standard errors, 2.56 standard errors will be 2.56 into 0.06, which will be equal to, I think I'll just use my phone calculator for now. 2.56 into 0.06, 0.1536. Yeah, so you have mean plus 0.1536 and mean minus 0.1536. So 4.8 plus or minus 1.536 will give you 99% confidence interval. So that is what you should be reporting as your this thing, lower and upper limit. I think you should report it with two decimal places. instead of 4 decimal places as such. So I think 4.8 minus 1.536, is it right? Sorry, 0.1536 gives you around 4.6464, I think. Yeah. Yeah, almost this would be the correct answer. Dakshata, please repeat this question. Okay. So the last three, focus on the last two questions, questions 15 and 16. So what they are asking you to do is they are asking you to find the lower and upper limits of ln lower and upper limits of x with a 99% confidence interval with 2 decimal places. So what you want to do is you first have to keep this fact in mind that 99% confidence interval almost signifies 2.56 standard errors around mean, standard deviations or standard errors around mean, and your standard error would be around 0.06. So what you should do is you must calculate 2.56 times the standard error, which will give you a value. So your mean, which is like 4.8 plus or minus your 0.1536 value, upper limit 4.95 is correct, sir. Yeah, I think it will be correct. 4.8 plus 1.1536, right? So I think it will be 4.9536. Can you guys see my screen actually, like now? Uh, Anyone? Can you reply? So I think that's all about for the questions. Like I think we, even though I didn't cover like the last 5 questions, I think we covered that through discussions as well. And I think someone here asked about how to address Z table and T table. And Ranu Hiral Yadav has asked how to question 1 and 2 again. Are you guys here still? Like Ranu Hiral Yadav and
Yes, I'm here.
yeah. So questions 1 and 2 of week 7, right?
Yes.
Yeah. Okay. So questions 1 and 2 are pretty simple. So they are saying that so given data to you is that you have read a paper, and then a paper, some 32% of the bacteria are virulent and others are benign, and then you do the same experiment yourself, and then you find that 76 of 200 bacteria are virulent. So using your data, whatever you have performed, they're asking whether you can support the paper's data, which is like 32%. If you see 76 by 200, it gives you 0.38, but here it's given 0.32, which is 32%. So both of them are not same. So they're just asking if you can support your data, so support their data with your data with a 95% level of confidence. So what we proceed with this problem using measurement of proportion, and using that, what we usually do is we take this, we just write down our values. So p value would be 0.32, 1 minus p value would be 0.68, and your 76 by 200, 76 bacteria out of 200 bacteria virulent will be 0.38. So what you do now is you form an null hypothesis saying that your p-value, which is that the number of virulent bacteria, weighted probability of all the virulent bacteria, is less than 0.32. and you perform your statistical tests to find whether your probability of this null hypothesis is actually greater than 95%. If not, you will reject your null hypothesis and accept an alternate hypothesis saying that your p-value is greater than 0.32, like that is which is 0.38, like 0.38 is greater than 0.32, right? So you can accept your alternate hypothesis and you can give yes as an answer. So null hypothesis p is less than 0.32. So before proceeding with the standard deviation, you must whether first find out whether your given data will give you a normal distribution. So for that, what you do is you multiply p and 1 minus p with the number of samples 200, and then you can find out what normally you can find out that both of them are greater than 10. This is 64 and this is 136, which is above greater than 10. So this will give you a normal distribution. After that, what you do is you try to find out the standard deviation. Standard deviation using this formula, p into 1 minus p divided by n, and this will give you the value of the whole root will give you a value of 0.03298. And then using this value, standard deviation, you try to find out the z score. Your z score will be your expected your measured p value minus the observed p value divided by the standard deviation, which will give you 1.82 z score, that is 1.82 standard deviations. So now you have to find out whether your whatever data you have obtained here through your experiment, that is within the 95% level of confidence. 95% of level of confidence gives you 1.96 standard deviations, but here we have 1.82 standard deviations. So whatever data you have, that is within the confidence interval of 95% only. So you can directly say yes. You can say that yes, I can support my paper. Alternatively, this is a shortcut method. Alternatively, through a long shot, what you can do is you can try to find out the area using the Z table for 1.82 Z score, and you can try to find out the probability using the area under the Z curve, and which will give you 93.12% probability. The do you get this question, like question one?
Yes, yes. I understood, sir. Thank you.
Yeah, Z score you already calculated here, so you just plug that into the second question and then you will get 1.82. Okay. I'll come back to the chat box. Dakshata Radhakar, question 15 is 16.5, question 16 is 68.5. I don't think your lower limit and upper limit would be that much varying, Dakshata, because your standard deviation itself is very low, right? Your standard deviation is in decimal places only, and if you multiply that by 2.56, I don't think you would be getting that much of a variation in your lower limit and upper limit. So I think you have made a mistake. Instead of maybe I think you might have made a mistake somewhere. So someone here asked how to utilize the Z table for finding the Z values. So I'll just take this as an example. So we have a Z score of 1.82. So using that, we'll try to find the area of the curve, area of a normal distribution curve uh, with a Z score of 1.82. So I'll go back to the Z table that we have posted later. Yeah. Here I have Z tables for Z value greater than the mean and Z value less than the mean. So we have a Z value of 1.82. So what we can do is Z value is greater than 0 here. 1.82 is greater than 0. So we'll try to find the area of the curve under this. So what you can do is your value is 1.82. So you take the first two base values 1.8. So Z will be 1.8 and then the precision value will be 0.2 here. So under 1.8, try to find out 0.2. So you get 0.9656. So that will be the area of the curve under with the Z value of area of the normal distribution under a z value of 1.82, and this is the area, and let's just say this is 1.82. So this area, this shaded area will be 0.9656. Yeah. So to find out the probability or the confidence interval around this, what you will do is you will subtract this by 0.5. So what happens is whatever area that is to the left of the this zero line, it will go away, and then you multiply this area by 2. You subtract this whole area by 0.5, and you multiply again the shaded area into 2, so that you will get plus z. You will get an area which is bound by plus z and minus z. That will give you the confidence interval or the probability. I hope I'm clear with the utilization of Z tables. T table is also the same. So instead of Z table, let's just say that we have to use a T table. So in T table, what kind of parameters do we need is we need degrees of freedom, and then we need a confidence interval. So degrees of freedom, say that we have 10 observations. 10 observations, we can't use a Z table because it's less than 25. So we have to resort to a T table. So to find the degrees of freedom, I'll just type in the degrees of freedom will be n, your number of observations minus 1. So degrees of freedom will be, if I have 10, if n is equal to 10, then your degrees of freedom will be 9. Yeah. So let's just say we want to find the t value of 9 degrees of freedom under 0.01 confidence interval. Yeah, for a two-tailed t-test, two table. So we have degrees of freedom of 9, and under 9 we have to find out under 0.01. We are taking two tails, we are not taking one tail, because t curve is same as the z curve. Right now we are assuming that the t curve is same as the z curve, so we are taking two tails. So under 9 and under 0.01 we are getting 3.250 is your t value. So let's just say you get a t score or a z score of 1.82 and you have 3.250. 1.82 obviously lies within 3.250. So your whatever data point you have, that also lies within your selected confidence interval distribution. So you can confidently say with that amount of confidence that your experiment, your null hypothesis is accepted or rejected. I hope everyone is clear with the usage of Z table and T table. So I'll leave the call open for another 7 minutes. So till like 4:50, and if anyone has any doubts, they can shoot it. Else we'll conclude the session. Please repeat about question number 11. Question number 11 in week 7, right? Yeah, question number 11 is pretty simple only. So you have x and y, you have their mean, standard deviations, and number of data points. They are asking you to, like, they are given a scenario where you are multiplying x and y, and then you are getting a plus or minus b. Your a will be the mean value, and then the b will be the error value. So that's what, and in this question, 11th question, they are asking the a value, which will be the mean of x and y, xy. So what you do is you just simply multiply the mean of x and mean of y. So 4.8 into 6.6, it will give you 31.68. Error value, you do the same thing. So error value, we discussed earlier that you have to if in case of multiplication, you have to find the percentage values, the relative values, not the absolute actual values. So your delta z by z, the error value in z divided by z, will be equal to the square and root of delta x by x the whole square plus delta y by y the whole square. Okay. Dakshata, I can go ahead.
Sir, question number 13.
Can you tell me what question number 13 is? Ln x.
Ln x. Yes.
So ln x will
How
be
it calculate?
ln x will be the derivative. So you have to find the derivative of the curve under the x under the x bar, and then you have to multiply it with the error of x.
Sir, can you repeat this?
So ln x will be the same as your multiplication stuff. So to find out the error, you have to find the uh, e- dou by dou x, and then dou z by dou z by dou x, and then you have to multiply it by the error in x. What is the significance of ln x? I mean, ln x is just a normal mathematical function. It's a natural logarithm of x. So ln x is used in many function, like many situations to get something out of x. It's just a mathematical function. So like in many, I think in many cases, like if you are from a biological background, then you can say that many models can be model many population growth models involve the use of logarithms. Sometimes natural, sometimes normal logarithm. Gupta, you can go ahead. Yeah, yeah.
Sir, I am asking about this law in respect of question number 13.
Okay. 13. Ln x is equal to a into b.
So, is there any same meaning log x of natural log x, or is it different there?
I mean, if you are going through a scientific calculator, then both of them will give you the same thing only, right? Like,
No, in case of if we are writing like base, so it means log 10x. But if we are taking ln, so it means log natural of
yeah,
x.
yeah, natural log of x.
So it is base e.
It'll be log base e only. So it'll be like 2.303 into
So in
that log
question
10.
number 13, or what I have asked here is the same thing, sir.
Yeah.
Okay.
Only thing is that they're
That's
asking the in question number 13 and 14, they're asking the actual x
the
bar,
natural log
yeah, x bar
of x.
value. Yeah, ln x bar. In the 14th question, they're asking the error value.
So we should take the log of this mean value, sir, 4.8,
Uh,
or what should we do in this case?
I like you have to take so ln is basically 2.303 into log base 10 x, right? So normally your log will be in the base 10. So your log, whatever you're taking log of x, that will be in the base 10. So
So
if you
that is written is like log, sir. In that case, so
yeah.
the
So if
base
you
is
want to
also
find
given
so if
log
you want
10x.
so if you want to find ln will be 2.303 log base 10 x. Ln x will be 2.303 or something, some constant. I think I'm not sure if it's 2.303 or something. So ln x will be 2.303 log base 10 of x. So whatever value, so if it's already, if you can find log
Oh,
only as well, but multiply that by 2.03.
right,
Many
sir.
many people won't know this formula, but in your calculators, in your scientific calculators, in your computer calculators, you will also have this ln option as well.
Right, sir. Right, sir. Thank you, sir. Thank you a lot.
Yeah. So all right then. So if we don't have any further doubts from this, like we'll conclude the session. And yeah, once again, thank you all for coming to this session. If you have any other doubts with respect to this session, you can feel free to ask these questions in the Google Groups discussion forum as well. Amit Srivastava, unfortunately, I actually can't share my number, but this you can contact me anytime through the Google Groups forum. I'll be answering questions there. Yeah, yes, yeah. So if you have any doubts, feel free to ask it in the Google Groups forum. And for those who actually can't view this video will be recorded and it will be uploaded in my YouTube channel. So this video will also be posted in the problem solving assessment session of your NPTEL course section. So you can go over. I think PPT will also be shared in the problem solving assessment. Will you share PPT today? Uh, Today maybe not. I'll try to upload the materials by Monday so that you can go over it. So apart from that, yes. So I think this documents will be shared in that problem solving assessment session tab in your course section. So whenever you want, you can go there and access it. This and the video as well. So I think that's about it. Being a chemistry background, this mathematical problem is very tough for me. So I don't think this problems like yes, except for this median kind of stuff. So I think except for this median kind of stuff, I don't think any other problem need to be solved by hand. You can always take a calculator, like you have a scientific calculator in your computer as well, which you will be accessing in the exam as well. So you can try to make use of the calculator, man, which will, I think it will help you solve 95% of the problems. Only for this median, mean, and all the stuff, if you, okay. Yeah, only for median and all the stuff, if you know the formula at the right time, I think you can clear it off. Yeah. So, okay then, I thank you once again for coming to this session, and I'll see you in the next class, 16th March, next Saturday from 3 p.m. to 5 p.m. Okay then. Yeah.
Thank you, sir.